Hello, everyone, and welcome to the second CHANGER Café. Today's topic is ethics of digital images in research. I am Antonija Mijatović. I'm a postdoc at the University of Split School of Medicine, and my background is in biophysics, but I do research that is interdisciplinary. It involves research ethics, also ethics of emerging technologies, metadata research, and also a little bit of artificial intelligence. Before we start, I have a question for you. So look closely at this image, and without having to know anything about Western blots, for example, this is a Western blot, without having to know how to interpret this image, just look at the visual cues and write down in the chat window, do you think that this image has been inappropriately manipulated? Yes or no? Search for the visual cues. So let's say you are a reviewer or you are a reader of a scientific article, and then you find this image, this scientific image published. Do you find anything suspicious? The first block has 4 signs, while the second has 5. If you think on these pluses and minuses, you don't need to look for those. You only look for the blots within the image. If you look closely at the lanes 2 and 3, these 2 bands are completely the same. They are duplicated. And you can also look for more because this is not the only duplication. Is it an image about cell signaling? Well, this is a Western blot. I'm not an expert for this type of images, so I'm not sure, but that's not the point. The thing is, you don't need to know how to interpret these images. You don't need to be an expert in microbiology, in histology, anything. You can just look for the visual cues in order to search for duplications. So is it cell signaling? Is it protein expression? I don't know. But what I can tell is that this image contains duplications. So let's see the answer. So the first one was found by Wiki. But then if you look at the second row, you'll notice that these blots are also completely the same. They have been duplicated. And some of them are even stretched out, so they don't seem completely the same. However, if you look closely, and also if you analyze it with a specialized tool, which I'll mention later in the presentation, you will see that they are copied and pasted. So this was an image duplication, and this is considered an inappropriate image duplication because editing scientific images in order to make them more clear is completely fine. However, purposeful image manipulation, which aims to distort scientific experiment results, is considered a severe research misconduct. And aside of image duplications, there are other techniques, such as cropping important parts of an image, also color adjustment and selective enhancement. For example, putting too much brightness in the picture so that some parts of an image get invisible. Also, putting objects within the image that were previously not present and deleting certain part of an image. So today our focus will be image duplications because they are the most easy to detect with naked eye, because if you want to know whether something has been deleted, then you need to use specialized tools. It's not possible with naked eye. But before we dive in, and I have another question for you. Again, write in the chat window. What do you think? What percentage of published scientific articles do you think contain inappropriate image duplications? Of course, you don't know the answer if you haven't studied about these topics, but just lucky guess. Okay, 20%. Wow, that's a lot. Christos, even more, 30%. Wow. What do others think? What do others think? Is it less than 20% or more than 30%? So you agree with Vicky and Christos that it's around 20-30%. If true, that would be a disaster because then we wouldn't be able to trust science anymore because 30% would be a lot. After seeing the previous picture, duplication, I got suspicious. Tina thinks it's around 10%. Okay, others, do you think it's less than 10% or more than 10%? But yes, I agree, we should be suspicious. Because the image I showed you was published in a research, it was published in a scientific journal. So it wasn't manipulated by us. We didn't synthesize that picture. We found it in a scientific article. Okay, so the answer is 4%. Okay. And this is already a lot because here we are talking about duplication alone. These are manipulations that are possible to detect with naked eye. And it's only image manipulation, not data fabrication, for example, data fabricated and shown in tables, graphs, etc. So only image duplication visible by naked eye is quite prevalent, much more than previously thought. And studies show that approximately 4% of papers contain some form of image duplication or manipulation. And that's concerning. So when it comes to image duplications, there are three major types. First, there are simple duplications. So if you look at the example here, is the same image that appears multiple times in a research article. And that could be a result of an honest error. For example, a researcher didn't label the pictures correctly, and it happened that the same picture appeared twice in an article. So that's an honest error, but still it's something that shouldn't happen because this means that the researchers were very sloppy in their work. Something more serious, duplications with repositioning. This means that there are areas of image that appear multiple times, but they are rotated, zoomed in, or zoomed out, so that it's not possible to detect immediately. And this, well, could there be honest error? Well, depending on the way how images are produced, but they probably are a result of purposeful image manipulation. And then the third type, which is the worst, is duplication within the same figure. So if you look at the image here, again, we have some Western blots, and there are three duplications within the same figure. So what do you think? Could this be an honest error, or can this be a proof of data fabrication and research misconduct? So this most certainly is purposeful. You can't do this. It cannot be an honest error. And this, unfortunately, this graph was also published in a scientific journal. So now that you know more about this topic, now that you've seen image duplications and that you know what types of duplications there are, I have a challenge for you. I will show you three images, and all of those were published in research articles, and all of them contain at least one duplication. So let's look at the first image. This is composed of 4 images. And these 4 images represent 4 experiments. Therefore, they should be unique. There shouldn't be a part of an image that reappears in another image. So let's say that the images in the first row are 1 and 2. Images in the second row are 3 and 4. Feel free to write in the chat where is the duplicated part. which images contain duplication. So the first image contains a part that has been duplicated somewhere else. And this is the answer. So images 1 and 3, if you look at these parts, they are completely the same. Okay, now the second image. This one is a little bit more difficult. Here we have 8 experiments, and there are more duplications, not just one. How many duplications do you find? There are 3 duplications, because if you look at the first and second image, these areas, if you look at the clusters, they are completely the same. So are the areas on the first bottom images. And we also have another duplication here in the right. So if you remember the classification of duplications, these are duplications with repositioning, and they are harder to detect compared to simple duplications. And now let's move to our last example. Again, we have Western blots. Write down how many duplications you find. So you compare each blot with the rest of the blots and write down how many you find are duplicated. If you remember, we have duplications with repositioning, so sometimes they are larger, smaller, longer, etc. So they don't need to be exactly the same. The size of the blots can vary, which can make the duplication detection even more difficult. Okay, so majority of you found 2 duplications, and that's great because, as I mentioned, even if you found 1, that's an indicator of research misconduct and can indicate that the results are fabricated. So let's see how many duplications we have, because you will not believe it. So if you look at the second blot, it has been copied 4 times. Okay, then we have this blot duplicated twice. This blot appears 3 times. The blue one appears 3 times. And lastly, the red one appears 3 times. So this whole image is practically fabricated, and this is the worst type of image duplication. It's duplication within the same image. So can we trust this article? Can we trust this data? Absolutely not. Did you find this task challenging or difficult? So how do we detect? By visual inspection. We are looking for repeating patterns, we are looking for irregularities, we are looking for inconsistencies. For example, those inconsistencies can happen in the lightening and shadows, but you notice this is really difficult and it requires a very trained eye. And I would say not just a trained eye, but it requires a special talent, because personally, I find this task really difficult. No matter how many images I look, no matter how much I know about this topic, for me, this is really difficult. Luckily, we do have software tools that can do this automatically for us. There are tools that are based on the traditional photoforensics, and these include programs such as ImageJ, Forensically, Photoforensics, and even Photoshop has a feature called error level analysis. And the good thing about these tools is that some of them are free, such as Forensically and Photoforensics. You can search them on the web, and you can upload a picture that you find suspicious, and you can analyze it completely for free. And they are quite accurate. There are also AI-based tools. They employ machine learning, and they were trained with a large data set in order to spot image tampering. And first, let's see about photoforensics tools. So how do they work? These tools analyze pixel level data, and they also look at the metadata of the images. And they search for inconsistencies that were introduced during picture editing. For example, we have a very common technique that's called error level analysis. And this technique detects differences in compression levels across an image. Because if you look at the unedited areas of an image, these areas have consistent error levels. However, areas of an image that have been manipulated, they show different error patterns, and they will look differently under the error level analysis filter. So this is why this tool is very useful when searching for the duplications, but also searching other manipulations that are not possible to be detected with naked eye. For example, areas that have been added later in the image or areas that have been deleted. However, this tool only works for JPEG images. It doesn't work on the PNG images. And the reason is because the JPEG leaves compression artifacts. So each time an image is edited and resaved, there are artifacts left, and they can be seen with ELA tool. Also, the image metadata, each time an image is resaved, the quality of the JPEG drops, and that is something that can be seen in the metadata. So how does this tool work? So let's look at this very beautiful image. We can see ruins of an old Greek temple, and we can see a full moon. And even with our naked eye, we can tell immediately that the moon has been added. It's an object that has been added later. But what about the ruins? What about the temple? Is it fake? Is it real? Has it been edited? It's hard to tell. So let's apply ELA, error level analysis. And when applying this tool, this is what we get. So if you notice, the area of the moon looks differently compared to the rest of the image, while the temple looks the same. So we can conclude that that the moon is an object that has been later added to the picture. But if we look closely at the columns of the temple, we can see a little bit of editing. Probably some shadows and light have been added later, but the ruins they were present in the original image. When it comes to clone detection or duplication detection, if you go to photoforensics tools, you can use this feature, and it will highlight areas that have similar regions. So regions that are similar are marked in blue, and they are connected with a red line. This is if you have multiple pairs of duplication, so you know how they are paired. So this is also a good indicator that the image has been manipulated. However, pay attention that if you upload a suspicious image to this tool, it won't give you an answer immediately, but you need to play with this tool. You need to set image region and size of the areas you want to compare. So it's not very easy to use. So these tools have limitations. Filters like ELA only work on JPEG images, and they cannot be used for other formats that are popular, such as PNG. And also, these tools will not tell you where manipulation is, what type of manipulation there is, but you need to know how to interpret the results. So these are prone to human error. And also, without the access to the original file, it's hard to confirm what had been altered. Also, using these tools is quite time-consuming, and especially if you're dealing with large data sets. And finally, these tools cannot detect AI-generated content, such as synthetic images and deep fakes. So speaking of deep fakes, I have a new challenge for you. So I will show you images of 6 people and write down in the chat, who do you think is real and who was created by artificial intelligence? Now let's see the answers. And the answer is, they are all AI generated. So none of them is real. They have been created using deep learning model. And if you go to this website, thispersondoesn'texist.com, each time you refresh the page, you will get a new image of a person who doesn't exist. So all of them were created using this deepfake image generator. But let's look again. So you were able to tell that some of these were created with AI. For example, if you look image 4, person 4, you will notice that she has different earrings. So this can be an indicator that it is an AI-generated image. Also, if you look at the backgrounds, for example, for image 4, image 2 even, image 3, then the backgrounds are quite abstract. So that can also be an indicator of AI-generated images. However, it's really hard to tell for person 1, person 5, person 6. And these tools are getting better and better, so they will make less and less mistakes. So how do we detect these images? And also, AI-generated images appear in science as well. So they appear to show experimental results, but they may not be based on real data, and that is very concerning for scientific research. So these are very difficult to detect, and even for trained experts. So let's look another example. I will show you 6 images of scientific images, and 3 of them are real and 3 of them are AI generated. Can you tell which of these 6 images are AI generated? It's 3 of them. And these are the answers. So images B, C, and F have been created with generative AI technologies. So how do we detect AI-generated images if we can't do it with naked eye? We also can't apply traditional photoforensics tools. We can only detect these images by using deep learning-based methods. So these are very sophisticated neural networks that learn how to detect inconsistencies or also anomalies in images, and they analyze visual features at multiple levels, so low levels and mid levels. And they have greater accuracy, and they also work with different formats, so not just JPEG, but also PNG, etc. However, the challenge with these tools is that in order to develop an accurate AI tool, you need to train them on a really large data sets. So let's see a classical example of a neural network that can be used for this type of task. So we have convolutional neural networks, and without going into too much technical details, I'll just say that they analyze low and mid-level features of an image. Low levels features are pixels and also metadata. So those are the features that were analyzed by traditional photoforensics tools. But they also go one step further. They analyze mid-level features, and by this I mean objects of the image, for example, edges, textures, lighting, etc. And they search for inconsistencies in lighting or unnatural boundaries or texture mismatches, etc. There are different types of networks. So aside of convolutional neural networks, we also have Siamese networks. For example, these are architecture that are made of 2 or even more subnetworks. These subnetworks share the same weights and same parameters, and they process different patches of an image. And then when the outputs are compared, Euclidean or cosine distance is used. So this method, it's very effective when it comes to duplication detection. When it comes to tools that are used for AI detection, first, if you Google, you will encounter many free tools, many free online tools, such as AI or Not, Illuminati, etc. But the reason I didn't put these in the presentation is because their accuracy is really low. Their accuracy is around 60%, which is just about 50%, which is based on coincidence. So in order for these models to work well, they need to use advanced deep learning networks, and they need to be trained on a large data sets. So these are some of the tools that are used for duplication detection. For example, Proofig is used by publishers. It's used for image detection. For duplication detection, it has an accuracy, it's about 80%. We also have Image Twin that is used for manipulation detection, but also for plagiarism detection. I believe we have some colleagues from Image Twin who can later tell us more about this tool in the discussion section. Then Microsoft Video also has real-time video and image manipulation detection, and it is used by general public and content platforms. There's Hive AI detector, which is used by businesses and content platforms. There is Sensity AI, which is used by governments, by businesses, and also Adobe has its tool, which is used by creative professionals and publishers as well. So to summarize, image manipulation is more prevalent than previously thought. Approximately 4% of the articles contain some forms of image duplications. And traditional detection tools that are based on photo forensics are useful, but they are limited, especially when it comes to the format of the image. And also one needs to know how to use these tools, how to interpret their results. And another problem that's becoming prevalent are AI-generated images and deepfakes. They are also they are becoming more prevalent in science, and they are hard to detect. We can't use naked eye to detect these images, and we can't rely on traditional photo forensics tools. So we need to develop advanced deep learning models that are more accurate. But finally, we should prevent image manipulation from happening in the first place. And how to achieve this? There should be clear guidelines on what is appropriate image editing and what is inappropriate image editing or image manipulation. However, people who are not honest probably know that they are inappropriately manipulating images. Therefore, it is important to enforce consequences and penalties for intentional manipulation. And finally, we should promote a culture of integrity. So thank you very much. If you have any questions, feel free to ask now. If you want to share with us your personal experience with using AI tools or developing AI tools, also feel free to speak. If you remember something later, then feel free to email me at antonia.mijatović@mevsky. Thank you very much for your attention.